Hello students, this is Sheila Samuel. Our next topic is on Androecium, the male reproductive structure. Here you can just see the stamen. It is also known as microsporangium, and inside that you'll come across the pollen grains. So it's also known as stamen, and it has got two parts, filaments and anther. If you just have a look at the picture there, you can see the stamens also there. That is, it has got anther and filament, that is, it's also known as the stalk, and the length and the number of the stamens that always varies. Then you can just see the structure of the anther. It is mostly, that is, in this hibiscus we have come across monothecous type of the stamens, but here usually we'll come across bilobe dithecous type. That is, each anther will have lobes, and this each lobe will be having provided with two theca. That is why it is known as the dithecous. And if you just see the transverse section, you can just see that this anther has got four, that is, two lobes one side, two lobes on the other side. Overall you can just see that there will be four pollen sacs, and this four pollen sacs we call it as the microsporangia also. Then you can just see the line of descens there, and then you can also see the pollen grains there. That is, in the each four, that is microsporangia, you will come across many pollen grains. Now this anther, that is, it is a four-sided structure, and you can just see four microsporangia, and this microsporangia, that is, they give rise to the pollen sac also, or you can call it as the pollen sac also. Now let us see the structure of the microsporangium. That is, when you see the transverse section of it, it is circular in outline. That is, transverse section of the microsporangium, it is circular in outline. Now this microsporangium, when you are seeing the transverse section of it, it is surrounded by four layers. That is, outer one epidermis, endothecium, middle layers, and the tapetum. You can just see the structure also when it is it's being drawn, that is circular in outline. The center you can see there is a connective also, and here this is the transverse section of it. You can just see the epidermis, then endothecium, then you can find some layers, that is middle layers, and inside it you find that sporogenous tissues, or we call it as the microspore mother cells, and in that you can see that another layer, that is the, in the sporogenous tissue, that is the tapetum layer. Now here this epidermis, endothecium, middle layers, their function is to protect, and they helps in the descent of anther to release the pollen. Now tapetum layer, that its function is to nourish the developing pollen grains. If you just see the features of the tapetum cells, you can see that the cells of the tapetum, they have got dense cytoplasm, and they have more than one nucleus. Now microsporangium, if you just see inside it, you find the sporogenous tissue, and the sporogenous tissue, they are nothing but they later on mature to give rise to the microspore mother cells, and this microspore mother cells undergo meiosis, and each that is, when they undergo meiosis, they will form microspore tetrad. That is, in each microsporangium, that is, you will come across many such microtetrad. Here one microspore, that is microspore mother cell, is giving rise to microspore tetrad, means four microspore. There will be hundreds of microspore mother cells. They will be giving rise to so many such microspores, and these microspores, they are haploid, and we call it as the pollen grains, and this later on give rise to the male gametophyte. So here, and if you just see the structure of the pollen grain, the pollen, and you can just have a look when it gets dehisced, that is, when it is being, the pollen grains are liberated, you can just see that each microsporangium, and this microsporangium, so many microspores are present, and each microspore mother cells give rise to what? Four microspores. Now next you see, these pollen grains, they are spherical in spherical in shape and about 25 to 50 micrometers in diameter, and they are two-layered, XII and intine. XII has got different patterns and designs. It is made up of sporopollenin and it is a resistant organic material. It can withstand high temperatures and strong acids, and no enzymes can degrade it. And in some places of the XII, you'll come across some that is incomplete, is not continuous, that is the germ pores are present, that is where sporopollenin is absent, and this our pollen grains are well preserved. And you'll find that another layer, that is the intine. Intine is thin cellulose and it is made up of cellulose, pectin, and it is a continuous layer. Pollen grains, when they mature, they will form two types of cells, that is the vegetative cells and the generative cells. Vegetative cells, they are bigger, food reserves, that is in large number, and they have got an irregular shaped nucleus. Generative cells, they are small, they floats in the cytoplasm of the vegetative cell, they are spindle shaped, and they have got dense cytoplasm and nucleus. And when this, that is 60% of the angiosperm, they shed the pollen grains at two cell stage, that is they'll have a vegetative cell and the generative cell. But remaining species, they shed the pollen grains at three cell stage, that is the vegetative cell and the generative cell. Generative cell will give rise to two male gametes. So including these two male gametes in the vegetative cell, we say that it has been shed at the three cell stage. And this pollen grains, for to many it is allergic, and the study of the pollen grains, we call it as the palynology. And there is one pollen kit also that is present in insect pollinated species. It is made up of carotenoids and lipids, and it has got a sticky layer which is been secreted by the XII, and this has been given out by the, that is secreted on the XII. You'll find it is a sticky layer. This is separated by tapetal cells, and here these also act as a insect attractant, and this help against UV. And this pollen grains, this is very rich in nutrients. They have been used as food supplements in the form of tablets, etc. Then you find that how long this pollen grains will be viable. So here you find that they are highly variable. They depends on prevailing temperature and the humidity. For examples, in cereals like rice and wheat, they lose their viability within 30 minutes of release. And in Rosaceae family, Leguminosae and Solanaceae, it may be viable for many months. And usually these pollen grains can be stored in liquid nitrogen, that is minus 196 degrees centigrade. They are used as the pollen sacs. And another thing to be noted is that when the pollen grains land on the stigma for fertilization, at that time how long they will be viable, that we have to see it. So this much under the microsporangium, that is the anther, and I hope that you have followed it. So next part, that is the female section, that will see it in the next video. And thank you so much.